Good day everyone, welcome back to Majek School. So under our metabolic pathway course, we'll be talking about nucleic acid today. So yeah, let's get into it. So nucleic acids are large molecules that are composed of monomers called nucleotides. So these nucleic acids can also be referred to as polynucleotides, which means they contain more than one nucleotide, a whole lot of number of nucleotides. Now these nucleic acids, they act as the transmitter or like the storage of genetic information of cells, tissue and an entire organism. So now these nucleic acids help to preserve the genetic information of an organism and transfer it down to the next generation. Now they are called nucleic acids because they were first discovered and as acidic medium in eukaryotic cells in the nucleus. But with recent findings, this nucleic acid has been discovered in the cytoplasm of eukaryotic cells and also in prokaryotic cells that have no nucleus. Now these nucleic acids, there are two of them we have in an organism. We have the DNA and the RNA. The DNA, which is the deoxyribonucleic acid, and then the RNA, which is the ribonucleic acid. The DNA can be found in the nucleus, can be found in the cytoplasm, and can also be found in the mitochondria. So now we have three types of RNA. We have the messenger RNA, we have the transfer RNA, and then we have the ribosomal RNA. Now these DNA and RNA, there are little differences between them. They are all composed of, both of them, the nucleic acids, RNA and DNA, they are both composed of nitrogenous base, sugar base, and a phosphate group. But now the difference between the DNA and RNA is DNA is a double strand helix, while RNA is a single strand. Now for DNA, the nitrogenous bases there are cytosine, guanine, adenine, and thymine. But now for RNA, we have uracil, cytosine, adenine, and guanine as the nitrogenous base. Now for DNA, we then have deoxydiribose sugar as the sugar base there for DNA, while for RNA we have just the D-ribose sugar. So those are the major differences between the RNA and the DNA. Now complete hydrolysis of nucleic acid will give nitrogenous base, sugared bases, and their phosphate group, because these are the components and the constituents of this nucleic acid. They are what make up this nucleic acid. Now something to also note is that the phosphate that is found in this nucleic acid is orthophosphate. Now let's consider the components of these nucleic acids, the nitrogenous base, the sugar base, and the phosphate group. So now we have two nitrogenous bases. We have the purines and the pyrimidines. What makes up the purine is a pyrimidine and an imidazole ring. These purines are derived from the compound purine, and purine is gotten from the fusion of pyrimidine and an imidazole ring, while pyrimidines are derived from pyrimidine, which is a heterocyclic compound. So pyrimidines are derived from the compound pyrimidine. An example of these pyrimidines we have are cytosine, uracil, and thymine. Now let's look at the numbering of these nitrogenous bases. So for purine, it goes in an anticlockwise direction, the numbering, and then for the pyrimidine, it goes in a clockwise direction. Now something to also notice is that these nitrogenous bases can exist in the keto and the enol form. To put it in simple terms, now the keto form is when carbonyl group that is there, but now when it's an enol form, is an hydroxy group that is there in the place of the oxygen. It's now hydroxy group that we have there instead of oxygen. All the pyrimidines can have this enol form, but only guanine can have the enol form among the purines. Adenine can't have an enol form because it has no oxygen in its ring. At the physiological pH, the keto forms are the predominant, more found compared to the enol forms. So now for the sugar base, as I said before, the DNA has the 2-deoxy-D-ribose sugar, while the RNA has the D-ribose sugar. Now this ribose sugar is a 5-carbon sugar which is folded into the furan ring. And now the ribose sugar, both the deoxyribose and the ribosine in the DNA and the RNA is of the beta conformation, which means when this ribose sugar has been arranged in the furan ring, arranged as a ring, the hydroxy group of the first carbon is up and not low. For the alpha, if it's an alpha conformation, the hydroxy group will be below. Now we call it D-ribose sugar because the D there represents or shows that is a enantiomer. which means it rotates plane polarized light, and it's D, which means it's texturorotatory, which means it rotates the plane of light to the right. Now, lastly, we have what we can call nucleotides and nucleosides. Now, nucleosides is the combination of just the nitrogenous base and the sugar base. So, example, we have the adenosine, guanosine, cytidine, and uridine. Now, for the pyrimidines, when there's a sugar base being attached to it, it gets an ending of idine, like idine. Why for purines, when they are attached to a sugar base, they get the ocene suffix at the end. Now, these nucleosides, when they are attached to a phosphate group, they then become nucleotides. And then in these nucleotides, you can have just a nucleotide that has one phosphate group, two phosphate group, or three phosphate groups. So examples of this, we have the AMP, we have the ATP, we have the ADP, we have the GTP, and the rest. So a nucleotide is made up of the nitrogenous base, the sugar base, and also the phosphate group. Now, the linkage between the sugar base and the nitrogenous base is a beta N glycosidic bond. For purines is N9 that binds to C1 of the sugar. While for pyrimidines is N1 of the pyrimidine ring that binds to C1 prime of the sugar ring. Phosphate group is attached to the sugar molecule by esterification of a definite hydroxy group of the ribose sugar. So that will be all for this video. Thank you for watching. Don't forget to like and subscribe. Thank you.